Good morning, everyone. Okay, welcome back. Just taking a few seconds here to get everybody logged in. Okay, all right. Well, good morning, everyone. I hope you're doing well this morning. And as you know, as we continue to race along towards the end of our semester. So no mystery here. You appreciate that this coming Tuesday, you will have your third and final lab exam of the semester. Once you complete your exam this coming Tuesday on class marker, stand up and have a good shout because you will have completed your lab curriculum and you will be finished with lab for the semester after this coming Tuesday. Okay, so give it a real hard push here and try to finish strong with the lab exam and you'll be all done with lab. Okay. Also, continue to remind you about your final exam. Okay, your final exam for this course is Tuesday, December 14th at 8 a.m. Tuesday, December 14th at 8 a.m. Okay, gang, do you have any questions right now about our schedule as we approach the end here? Okay, we are going to be getting back into chapter 15, and my goal is to finish coverage of chapter 15 today. That's an ambitious goal because there's a lot of content, but we're going to make that happen because I have to start chapter 16 next week. And next week is our final lecture week together, and I need 2 lectures to handle chapter 16. So do you have anything you'd like to ask at this time that you've been studying about the nervous system? Okay, well, if not, please remember you can always come visit me during office hours if there's something you're struggling with, okay? So without any further ado here, we're going to get started, get back into chapter 15, okay? So I'm going to share my screen with you. And the last time we were together, we were just getting started on this hypothetical scenario. As you look across the content of chapter 15, it would be terribly boring for me and you to just have me list out the facts of chapter 15. I mean, it's like reading an almanac. There's just anatomy after anatomy after anatomy after anatomy. And I don't think that's really what anybody's going to benefit from. So what I'm going to do here is continue with this scenario and try to connect the dots between certain pieces of anatomy and certain physiological concepts that we really want you to appreciate as you push through this content, because this is pretty dense stuff. Okay, so let me just read these notes to get us back into the mindset of what we're thinking about. Okay, so we're in chapter 15. Okay, and so I'm proposing this hypothetical scenario where you're getting scared. So you're sitting in a 4D movie auditorium. You're experiencing puffs of cold air at different moments of the movie. You can smell a gross odor in the room from equipment that's spraying a perfume into the auditorium. You see a scary image of Frankenstein walking towards you, and you can hear the sound of a werewolf in the background. Okay, so we're scared in this theater. And so what we're doing is processing, we're trying to understand the anatomy and physiology aspects of why we would be scared and how our body will respond. Okay, so, I'm sort of trying to demystify the physiological response of fear for you. So I'm taking away a little bit of the fun of going to a 4D movie. Okay, so obviously it all starts with your body receiving the stimulus, receiving the various kinds of stimuli that we're talking about here. So the first section of notes was the input. The input is always going to involve the sensory nervous system. And the sensory nervous system is basically the peripheral nervous system, right? I mean, there are some cases where you do have some internal receptors, but for our conversation right now, we are talking about the peripheral nervous system. So the first thing we mentioned was your eyes. Your eyes are looking at images of Frankenstein on the screen. So the photons of light coming off the screen are going to hit the back of your retina. That's going to generate graded impulses in the neurons of the retina, which will summate at the axon hillock and fire action potentials. Those nerve impulses will travel from the retina to the brain via the optic nerve, cranial nerve 2 that you've been studying for lab. We're going to come back to that in just a moment. Your ears are hearing the werewolf, so you're delivering information from the cochlea in the inner ear to ultimately the temporal lobe of your brain via the vestibulocochlear nerve. Your nose is delivering information from the gross aroma and fragrance in the room. That information encoded as an electrical impulse is ultimately going to the temporal lobe of the cerebral cortex via the olfactory nerve. Thermoreceptors embedded in your skin are detecting the coldness of the air that's being puffed onto the back of your neck. Neurons that are part of hair root plexuses around your hair follicles are generating electrical signals when the hair follicle moves, and the hair follicle is moving because wind, the puff of air, is blowing across the surface of your skin. Okay, so that's all from last time, and to set the continual stage for this morning's conversation. Okay, so we're getting the sensory information into our bodies through these various pieces of anatomy: eyes, ears, nose, nerve endings in our skin. Okay, so the next thing is, well, the brain itself is going to have to process this information so that we become consciously aware of what's going on and add an additional layer of emotion to that logical conclusion of what we're experiencing. And then the brain's going to have to decide what it's going to do about it. What is the brain going to do about the realization that we are experiencing these various kinds of sensory information? So the second stage of this process will be integration. Okay. And so, you know, if you've had any higher order mathematics like calculus courses and stuff, I'm not talking about integrals or anything like this. I'm talking about integration in the physiological context. Integration is going to be carried out in the central nervous system. So we're basically talking about your brain in the context of our current conversation here. And I'll even put up here for input for our conversation here, we're primarily talking about the peripheral nervous system, primarily the PNS, okay? All right, so all right, let me remind you of something, and then we're going to go back to the eyeballs as a way to think about what's happening. Okay, so what I want to remind you of, that, you know, obviously in the brain, there is a massive spider web connectedness of neurons. You have one neuron connected to another neuron, which is connected to another neuron. There's a chain of neurons that are connected to each other, okay? And typically between a sensory neuron and a motor neuron, you're going to typically have at least one bridging neuron. And those bridging neurons, you may recall from our time in chapter 12, are called interneurons. Okay, interneurons are all about the integration process. So neurons, networks of neurons communicate with each other through interneurons, okay, which act as bridges, if you will, or go-betweens or intermediary messengers, if you will, which act as bridges between one region of the central nervous system and another. Okay, so let's consider, as we appreciate that there's these things called interneurons, and their job is to facilitate communication between a sensory neuron and a motor neuron. Let's think about what that might mean in a bit more anatomical sense. So right now I'm going to go to visible body and talk about this from the eyeball perspective. Okay, so before we got started, I was loading up the software and organizing this picture for you so that we can have this conversation. So this is typically what you've been exposed to in lab, right? The cross section of the brain, and you're getting very comfortable with the internal anatomy of the brain at this time. And so, this is what we're looking at, okay? So what we're going to do is start our journey here at the eyeball, right? Your eyes are looking at the movie screen and you're seeing images of Frankenstein. Now, when the photons of light off the screen enter your eye, when those photons of light are in your eye, as they're just in your eye, your brain does not yet know that it's Frankenstein, right? Because the brain hasn't processed it yet. The processing of the electrical information that's generated from light entering the eye is the integration, folks. That's the integration. Okay, so, without being too insane here, what I'm going to suggest is the following, okay? That when we are looking at the movie screen and we see Frankenstein, okay, basically what's going to happen is photons of light are going to enter the eye, right? Photons of light are going to enter the eye, and at the back of the eye, along the border there, you have the retinal tissue. So the photons of light are going to continue till you hit these neurons back here. And from this point, the information content of the photons is going to be converted into electrical information. The fancy word in biology is called transduction. The information of the photons is going to be transduced into an alternative format. So you're going to go from light information to electrical information in the nervous system. So the light's going to hit the retinal neurons. They're going to depolarize, generate graded potentials, which will summate at the axon hillock to generate an action potential. And the action potential is going to fire and spread into the optic nerve, right? So this is all stage one in our notes. So where I've drawn these colored arrows is basically this part up here, the input, right? Here's the input moving along the optic nerve, okay? So as we continue the conversation here, we need to talk about, okay, so once the information starts to get through the optic nerve, it moves out into deeper areas of the brain so that the integration process can take place. And so what I'm suggesting now is that the nerve impulse, obviously, it's going up the optic nerve. It's going to cross over into the optic chiasm that you're familiar with. Okay, and it is going to go into the hypothalamus. Okay, it's going to go into the hypothalamus. And from here, what we're going to have is a collection of interneurons that are going to spread information to different regions of anatomy. So I'm going to color code an interneuron here as blue. What I'm saying is from the hypothalamus, you're going to have a synapse from the sensory neuron to an interneuron, which goes into, and I apologize, this isn't quite positioned perfectly, but you're going to have an interneuron branch off into the septal nucleus and go into the indusium griseum. Okay, And as you know, the inducium egregium is back here by the hippocampus, which is involved with memory. And there's also fibers that connect over to the amygdala, which are involved with emotions. Okay, so there's going to be an interneuron, and honestly, many interneurons that are branching off to the anatomy of your brain that is basically responsible for memory and emotion. The part of your brain that is responsible for memory and emotion is the limbic system. The limbic system has many pieces of anatomy, many of which you're being required to know for next week's lab exam. I'm simply not saying, okay, here's your limbic system, but let me show you what the limbic system anatomy is. So what I'm going to do is click the hippocampus, and then I'm going to ask the software to continue to categorize it till I get to the phrase limbic system. Now I'm going to select the limbic system anatomy. Here we go. 1, 2, 3, click. There, here's the limbic system anatomy. And I'm going to rotate this, even though it's going to disturb my drawing here. Okay. So the things that are color-coded blue right now by the software, that is all limbic system anatomy. Limbic system anatomy is memory and emotion anatomy. Memory and emotion anatomy in ways that honestly we're still trying to figure out. I mean, medical science is still trying to understand how this all works. Okay, so the limbic system anatomy is going to have information injected into it by an interneuron that connects with the optic neurons along the optic nerve. Okay, so I don't know if I can perfectly match that back up again, but this is the basic idea. Here we are. Okay, so at the same time that information is moving into the limbic system, because we're looking at Frankenstein on our movie screen, we're going to have other interneurons, and I'll make these guys purple here. We're going to have other interneurons that come off the optic nerve information as it passes through the hypothalamus. That other information is going to go into the back of the brain here at the occipital lobe, because the occipital lobe is responsible for logically and rationally processing the visual information. So what I'm saying is the information comes into your eyeball, the photons of light, you have electrical signals generated along the axons of the optic nerve. At some point along their pathway, typically in the hypothalamus, you're going to have interneurons which synapse with those neurons, and then boom, you spread all this information out, kind of like a wave spreading along multiple threads of a spider web simultaneously to different areas of your brain. Back here in the occipital lobe, you're going to have the occipital lobe neurons process that information and make a rational, and when I'm saying it that way intentionally, because I'm trying to tell you the occipital lobe is not, as far as we can tell, heavily involved in the creation of emotion or the retrieval of memory. The occipital lobe is heavily involved in the logical, rational realization of what it is you're looking at. So when you look at the movie screen and information goes to the occipital lobe neurons are saying, oh, that's Frankenstein. That's Frankenstein. I know that. Okay. So the occipital lobe makes a rational decision about what it is you're experiencing. And then there is going to be crosstalk, cross communication between the neurons of the occipital lobe and the anatomy of the limbic system. Okay, so there's going to be crosstalk here. So the rational decision, the rational realization that Frankenstein is on the screen is going to interact back and forth with electrical signals of the limbic system, and the limbic system is going to layer on top of your rational conclusion that you're looking at Frankenstein with that emotional aspect of, oh, Frankenstein's a scary guy. He's a big green guy, I guess, a big gray guy, and he could hurt me, right? I mean, this is a little bit idealized, but that's the basic idea. So the occipital lobe is giving you the logical conclusion that you are looking at Frankenstein without any emotional content. And then the crosstalk of electrical signals between interneurons of the neurons in the occipital lobe and neurons in the limbic system anatomy will be sure that you layer on top of that logical conclusion the emotionality of the experience that you are having, okay? And so truth be told, this is going to happen super fast, right? I mean, it happens basically faster than I can talk about it. It's happening so fast so that in a split second, you realize you're seeing Frankenstein, and in the next split second, you realize, oh, that's a scary thing, okay? So this is all happening terribly, terribly fast, not at the speed of light, but basically at the speed of electricity. So that's a little bit slower than the speed of light, okay? All right, so let me ask you a question in the chat window. Am I making sense right now, or have I lost you? Okay, all right. Thank you for the feedback. Okay, so it would be disingenuous of me. It would be dishonest of me to tell you that medical science knows exactly how this all works, because we don't. We don't. There are still gigantic research projects around the globe where neuroscientists are trying to really figure out all these synaptic connections and the pattern of behaviors that happen when we experience a single piece of stimulus, such as light from a screen. We don't know it all, and it's probably going to be a very long time, if ever, that we do. But I'm trying to give you this basic idea that your sensory machinery in the eyeball is generating electrical signals that inject electrical information that go up the optic nerve, across the optic chiasm, into the neurons of the hypothalamus, where they intersect with interneurons that then pulse that information out to different areas of the brain to be processed. So you have the rational decision that you're looking at Frankenstein from the occipital lobe anatomy, and the emotional awareness of how you should think about that coming from the limbic system. Once that realization is decided, then you're going to have other interneurons, and I'm kind of running out of colors here. You're going to have other interneurons that communicate information up into the post-central gyrus, right, which is where your body's map of itself exists. And those neurons are going to have other connections to the precentral gyrus, where you're then going to decide how you're going to respond to this realization that you're looking at scary Frankenstein. And let's be frank, if that was a pun, a bad one, let's be honest here. So you're going to have interneurons communicating with the post-central gyrus and subsequently the precentral gyrus. The precentral gyrus is going to make a decision about, how am I going to move my skeleton? How am I going to move my body when I'm looking at scary Frankenstein? And traditionally, what's going to happen is your precentral gyrus is going to coordinate the contraction of your skeletal muscle tissue, skeletal muscles in your legs, so that you contract them and you run away from Frankenstein, right? So you're going to have interneurons connecting up here. They're going to make a decision to run away. So that's going to then spread down through the thalamus, right? If I were to dissect away the forebrain segment, I would get to the thalamus, which is in this general central location of the brain, correct? So it's going to make a decision to go down here and out the spinal cord to contract your skeletal muscle tissue, and away we go. We're running away from Frankenstein, okay? again, this happens faster than we can possibly even talk about it and have a discussion. Okay, any thoughts, any questions right now? I mean, there's two takeaway messages, maybe more than that. All right, first off, you continue to appreciate that in this course, the content is overwhelmingly complex. That's the first thing. The second thing is, in the moments where you don't have to worry about getting tested on this stuff, you can step back and say, my God, this is amazing. How did anybody or anything ever create this? Well, folks, you're looking at millions of years of evolution right here, millions of years of evolution. Okay, so somehow this has all come together. All right. And then you have the third aspect of, holy crap, I'm expected to know something about this because I'm going to get tested on it. Okay, so that's what we're doing right now. Okay, any questions about this image before I go back to the notes? Okay, all right, so let's continue our conversation here. So this integration is happening in multiple areas of the brain, processing the same visual information differently. The occipital lobe, logical decision about what you're seeing, Frankenstein. Limbic system, what emotions and memory do I have associated with Frankenstein? Crosstalk of communication, overlay the emotion on the logical realization that you're looking at Frankenstein. Communication into the interneurons, into the postcentral gyrus, which has a map of where your body is in three-dimensional space right now. Communicate to the precentral gyrus. Let's make a strategy. Let's develop a strategy or plan of attack for how I should respond to this information. That plan of attack is, I'm going to run away because I'm scared of Frankenstein. So the decision is made in the precentral gyrus to send nerve impulses down the brainstem, down the spinal cord, out to the skeletal muscle in your legs to contract them, and away we go. We run in the opposite direction, and it happens, bam, just like that. Okay, so let me give you a couple more ideas here in your notes. The purpose of integration is to process stimuli so you can perceive the stimulus. So you can perceive the stimulus, and that means occipital lobe, I'm looking at Frankenstein. And experience the stimulus. By experience the stimulus, I mean add emotional intensity to your perception. Add emotional intensity to the perception. So that would be the limbic system anatomy. Followed by executing a plan of how to respond to that stimuli. And here's where we're going to involve the post-central and precentral gyruses. Okay, so, I've said this many times and I don't want to keep repeating myself, but this is the basic idea of what integration is all about that I tried to share with you in the visible body picture as we talked about our experience, our stimulation of seeing and experiencing Frankenstein and deciding to run away. Okay, that is what integration is. And this is going to happen in the anatomy typically of the brain. Okay, you can have a little bit of integration happen in the spinal cord with certain reflexes, but most integration is going to meaningfully happen in your brain. Okay, So please ask questions along the way if you have them, okay? And so let me continue here. I want to, we appreciate that from last time. I don't have my notes up from last time. From last time, I asked you to contribute some ideas about what you would experience when you are afraid. And many of you said, well, your heart rate would go up, you might sweat a little bit, you might feel anxiety, all kinds of different things that you said. We had a nice list. And so what we need to talk about now is the aspect of your response, and it's going to be a multi-layered, multi-pronged response of what your body experiences during the fear sensation, okay? And so, this is a difficult topic to deal with because there's so much that could be said, and all the things we're talking about basically are kind of happening simultaneously, because you're going to have multiple electrical signals spreading down multiple neurons down your spinal cord simultaneously, like multiple threads of a spider web out to different areas of your internal anatomy to coordinate the fear response. And so it's all going to start basically with the hypothalamus. Okay, you noticed with the visible body screen, I showed you that the information was coming in the eye through the optic nerve, crossing over in the optic chiasm and up into the hypothalamus. Well, a lot of responses to the realization that you're scared in the brain are going to be coordinated from nerve impulses that originate in the hypothalamus, because the hypothalamus is your main junction box, if you will, linking the nervous system to the endocrine system. So let's just take one quick peek here again. Okay, so I'm going to clear my drawing. 1, 2, 3, I'm going to clear it. I hope I'm going to clear it. Well, okay, it's not quite doing what I want. So ignore those drawings right now. But you appreciate that right here's your hypothalamus, okay? This is the junction control box. It's the intersection for much of the connectedness between the nervous system and the endocrine system. And that's how we're going to approach this topic moving forward, talking about how the nervous system then creates instructions that lead to the release of hormones that alter your physiology, alter what the internal anatomy of your body's doing, okay? So all the integration has happened in the brain, all these interneurons zipping signals all over the place. Then there's going to be decision to run away, and there's also going to be just a decision that to support running away, I need my heart rate to go up so I can run away. Okay, so there are going to be many electrical signals that come out of the hypothalamus, down the brainstem, out the spinal cord to various pieces of anatomy, which trigger hormonal release from glandular tissues. So let's document this in your notes. The master command and control junction linking the nervous system to the endocrine system is the hypothalamus. Okay. And so what we're going to go now to is the third stage of our activity. The first stage was input. We are detecting the sensory information, we are detecting the sensory stimuli, and we're bringing it into the brain. Once it's into the brain, we integrate it in complicated ways that even medical science truly doesn't understand very well yet. And then we need to respond. So the third aspect is the output, okay? So the output, once again, is going to be the peripheral nervous system, because these nerve fibers are going to branch off the spinal cord. So it's the peripheral nervous system. And if I'm being honest, we also have to include parts of the endocrine system. Even though I'm talking about nervous system functionality here in chapter 15, the reality of it is that the nervous system is going to trigger endocrine activity. So let me underline this. And let me back up for a moment and highlight something here. So all of this scary scenario in the 4D theater started with sensory information being injected into the body that required peripheral nervous system activity, right? Nerve fibers in your eyes, which extend off the brain. So that's peripheral nervous system. So the cranial nerves are peripheral nervous system anatomy, right? That's going to inject information into the brain. So the sensory information is using primarily the anatomy of the peripheral nervous system. The processing of what it is I'm seeing and how I should feel about it and what I should do about it is the integration aspect. That's going to be handled by the anatomy of the brain, the central nervous system. And then once the central nervous system decides, oh, I should run away, or oh, I should increase my heart rate, then the central nervous system's got to communicate back out the peripheral nervous system to get the body to respond as it wants to. Okay, so here we are talking about the output or the response. Okay. So let's talk about it this way. Okay. I can't possibly talk about all the responses you're going to experience. You remember that long list we had last time? We probably had 8, 9, 10 things on it. So that would mean 10 different conversations. I'm going to take two of them and try to describe them to you in some level of detail. So we're going to start here, and I'll do it this way. Response 1. Response 1. The hypothalamus responds by, sending nerve impulses down axons that are part of the sympathetic division of the autonomic nervous system. That's a lot of words there. So that takes us to a conversation where I want you to be looking at figure 15.2. Take a look at figure 15.2 in your textbook, okay? So sending nerve impulses down axons that are part of the sympathetic division. Here, figure 15.2 is critical. Sympathetic division of the autonomic nervous system. Okay, so there's some big figures in this chapter. And so I'm going to be asking you to dissect some of those figures in preparation for your final exam. By the way, question coming in the chat window. Have you noticed the final exam study guide posted in Wiley Plus? Have you seen it? Because it's supposed to be there. Okay, good, awesome. All right, so if you haven't seen it, please be aware on the lecture portion of Wiley Plus, the third lecture exam, which is your final exam, that study guide is now available to you. Okay, so continuing here. So one of the first responses that's going to happen as we realize that we're now scared, okay, is that the hypothalamus will send nerve impulses down axons that are part of the sympathetic division of the autonomic nervous system. That's figure 15.2. So I have another question for you coming in the chat window. Hold on a sec. This is a long-winded conversation here. And here we go. Using figure 15.2, what is the number of the vertebra from which an axon exits and travels to the suprarenal gland on top of the kidney? Give me the specific number and type of vertebra we're talking about here. Okay, so Lauren is first to respond. Let's give a couple more seconds because I want you to all look at this. Take a look at that figure 15.2. Let's get two more people confirming or disagreeing with Lauren. Okay, let's get at least one more person here. All right, so I really want you to stare at that. Okay, I really want you to stare at that and take a look. That there's pretty good consensus among your comments in the chat window, but not 100%. And so I will confirm with you. Take a really hard, close look. That is, the axon is coming out of vertebra, thoracic vertebra T10. T10. Take a real hard look at that. Okay. All right. So let's document this in your notes. Okay. Axons leave the spinal cord at thoracic vertebra T10. And again, you need to stare at figure 15.2 to confirm that, where these axons, where they synapse with a group of specialized cells contained in the adrenal gland, specifically the adrenal medulla portion of the adrenal gland, called chromaffin cells. Okay, so when I asked you to identify the suprarenal gland, that's the same thing as your adrenal gland. It's just a different way of saying it. So suprarenal and adrenal gland is the same thing. And as you can see, and maybe as you had already known, you have two kidneys, so you have two adrenal glands, one on top of each kidney, right? And so the axon leaves thoracic vertebra T10, goes out through a twisted, convoluted pathway, and it synapses with chromaffin cells located in the adrenal gland. Okay. Okay, so let me read this note to make sure I got it right for you. Axons leave the spinal cord at thoracic vertebrae T10, where they synapse with chromaffin cells located in the adrenal medulla of the adrenal gland, located on top of your kidneys. That is correct. Let's continue the conversation. Now, what I would ask you to do is go back a page in your textbook to figure 15.1. Go back to figure 15.1. Part of your study time at home, you're going to have to stare at figure 15.1 and make sure you appreciate the significance of figure 15.1. First off, I'll take note to you that if you look at figure 15.1, the first image there is panel A. It is the somatic nervous system. Okay, so look at that. The second image down is the first image of panel B. And for that, you're looking at the sympathetic nervous system. Okay. The middle panel is also sympathetic, and you notice here they are showing you the adrenal gland, and they're specifically showing you the chromaffin cells. That's what I need to talk to you about right now. So the chromaffin cells are being highlighted in figure 15.1. So as the axons that leave the spinal column at T10 vertebra synapse with the chromaffin cells, those axons will release, you can see it yourself, they will release acetylcholine. Okay, so acetylcholine is not just used for skeletal muscle contraction, it also triggers cells in the adrenal gland. Okay, so the axons that synapse with the chromaffin cells release acetylcholine. Okay, the acetylcholine depolarizes the chromaffin cells and triggers them to release two other hormones that I bet you have heard of, epinephrine and norepinephrine. Epinephrine and norepinephrine are basically chemical cousins, and this is basically what is meant when people talk about adrenaline being released. Okay, so the chromaffin cells are stimulated by the acetylcholine released from the axon leaving the T10 vertebra to release epinephrine and norepinephrine into the bloodstream. So let's think about what we've accomplished at this point, right? The imagery of Frankenstein's come into the optic nerve. We've integrated things in our brain, and the brain decided, okay, one thing we should do is get scared. So get scared means a whole bunch of different responses. One of those responses is going to be pump up the heart rate. Well, you probably know adrenaline is going to pump up the heart rate. So how is adrenaline provided? It is released from the adrenal gland. Well, why is it released from the adrenal gland? Because the nervous system is activating the release of the adrenaline through the synaptic connection of the nervous system to the glandular tissue of the adrenal gland. Okay, and that's what you're seeing in figure 15.1. Okay. Okay, and so again, this is all happening in the sympathetic division of the autonomic nervous system, because as you know, the sympathetic division is the fight or flight division. Okay, this is the division that prepares your body for war, so to speak, for threat response. So we now have epinephrine and norepinephrine in the bloodstream, and let's see, I'm going to highlight some phrases in the notes that you should definitely be paying attention to during your private study time. All right, so the epinephrine and norepinephrine from the adrenal gland was triggered by the neuron of the sympathetic division to release adrenaline into the bloodstream. So I'll just make a summary statement now about how your body's going to respond. Your body responds to these hormones. Okay, so I'm talking about epinephrine and norepinephrine by increasing heart rate and converting glycogen back into glucose. Okay, so in response to the epinephrine and norepinephrine, your heart rate's going to go up, and areas of your body that are storing glycogen, which are predominantly going to be liver and skeletal muscle tissue, are going to take that glycogen and convert it back to glucose. Why? Because glucose is the primary monosaccharide from chapter 2, which drives aerobic cellular respiration. And aerobic cellular respiration is all about creating ATP to power muscle contraction so you can run away. So basically, we are trying to ensure that we have a steady biochemical fuel supply to support the threat response that we want to execute. Okay, so your body responds to these hormones by increasing heart rate and converting glycogen back into glucose to ensure that muscle cells have a steady supply of biochemical fuel to support our fear response. Okay. I'll put in parentheses here a little subnote. So obviously the endocrine system through adrenaline is deeply important to modifying your heart rate when you're scared. I will also say that there are some other nerves that have other neurons that do innervate the heart that also modulate heart rate. We're just not talking about them right now because it's just too much. It's just too much. Okay. All right, so while you know a lot of the story at this moment, you don't quite know all of the story. Okay, and that's okay. That's okay. I'm not going to ask you about those things that we're not talking about. Okay, gang, do you have any questions right now? So what have we accomplished? I hope right now we have accomplished a basic understanding of how nervous system activity in the brain triggers nerve impulses in the hypothalamus, which then coordinate an endocrine tissue response. The adrenal glands are releasing epinephrine and norepinephrine into the bloodstream. Let's try another response. As I said previously, I was going to do 2 of these with you. Response 2. So at the same time that response 1 is happening, there are going to be other simultaneous responses happening. Okay, it's not like the body does one thing and once it finishes it, it does another thing. No, no, no. You're going to be doing multiple things at the same time. So simultaneously, with response 1, other neurons from the hypothalamus respond by, letter A, sending, so we're going to have different neurons coming out of a different section of the hypothalamus, sending down nerve impulses, again, of sympathetic subdivision neurons that synapse with cells that are part of the superior cervical ganglion. Okay, so take a look again at figure 15.2. Figure 15.2, take a look at the brain in the upper left corner of the brain. Slowly move your eyes down to where you see C1, C2, C3 and stop there. Now slowly move your eyes to the right. Do you see the superior cervical ganglion? The superior cervical ganglion. Okay, that's what I'm talking about. So the hypothalamus responds by sending nerve impulses down other axons, again, of the sympathetic division. I'm not going to keep saying this because this is all sympathetic division. sending nerve impulses down other axons that synapse with cells in the superior cervical ganglion. Okay, we recall, we know what a ganglion is. It's a group of cell bodies in the peripheral nervous system, right? So here you have an axon that's coming out of the hypothalamus down the brainstem into the first part of our spinal cord at the cervical region of our vertebrae, coming out and merging into a cluster of neuronal cell bodies that is named the superior cervical ganglion. Okay, so here at the superior cervical ganglion, the neuron coming off the spinal cord would be the presynaptic neuron. The presynaptic neuron is going to synapse with the cells that are part of this specific ganglion. And when they do so, the presynaptic neurons are going to release acetylcholine. Okay. At the synapse of the presynaptic cell to the cells of the ganglion, the presynaptic cell releases acetylcholine. Okay, so in the superior cervical ganglion region, we're having the release of acetylcholine from the neuron that comes down off of the brainstem from the hypothalamus. Okay. Now, what's going to happen is as follows. Okay. All right. So I'm going to tell you what's going to happen, and then I'm going to ask you in the chat window to tell me which of the pictures of figure 15.1 are we talking about. Okay. So I'm going to tell you what's going to happen. Okay. So, you're going to have the presynaptic cell generate, you're going to have the presynaptic cell release acetylcholine. That's going to activate the cells in the ganglion to do something. Those cells in the ganglion are going to generate an electrical signal that move out to a target organ. At the target organ, those cells that extended off the ganglion, so these are the postsynaptic cells, they are going to release norepinephrine. Okay, the postsynaptic cells of the ganglion generate an action potential from the acetylcholine that was released by the presynaptic neuron. Okay, that action potential spreads to its target tissue where the postsynaptic cell then releases norepinephrine. Okay, now if you look carefully at figure 15.2, there are a lot of axons coming off the superior cervical ganglion. Take a look at the superior cervical ganglion. There are a lot of axons coming off of it. And notice that those axons basically go up to your head. Some axons go to your eyes. Some axons go to the pineal gland. Others go to the lacrimal glands, you know, for tear production, that kind of thing, mucous membranes. What I want to highlight in my response at this moment is that you're going to have axons that innervate the glands that make up your salivary glands. That would be the parotid gland, the sublingual and submandibular glands. All of three of them are collectively your salivary glands. Parotid gland, sublingual, submandibular, these three glands are your salivary glands, okay? So the postsynaptic cells of the ganglion generate an action potential that spreads to the target organ where once that action potential reaches the end of the axon in the target organ, the postsynaptic cell releases norepinephrine, which is another neurotransmitter here, okay? And so here's what's going to happen. Okay. So the arterioles in the salivary glands, so remember, arterioles are tiny blood vessels. The arterioles in the salivary glands respond to the norepinephrine by constricting, by tightening up, by narrowing, by getting smaller in diameter, okay? The arterioles in the salivary glands constrict. When they constrict, they reduce the flow of blood into the salivary glands. Because the flow of blood into salivary glands is slowed down, we slow down our ability to actually manufacture saliva, because saliva, like all body fluids, is basically filtered blood plasma. So the arterioles respond to the norepinephrine that's released by the postsynaptic cells coming out of the ganglion. How do they respond? The arterioles constrict, slow down the flow of blood, so we don't make as much saliva. If we're not making as much saliva, we are going to experience dry mouth. If you've ever had dry mouth because you were nervous giving a public speech, this is what's happening. Your salivary glands cannot make the saliva because there's not enough blood running through those glands at that moment, because the blood vessels have narrowed. They've narrowed because the nervous system told them to narrow. Okay? Okay, so the arterioles in the salivary glands constrict, reducing the flow of blood into the salivary glands. You experience dry mouth. Okay. Whoops. Okay, so let me ask you a question in the chat window here. Okay, here I go. In figure 15.1, counting from top picture to bottom picture, which picture reflects the conversation of response to in your notes right now? Okay, in figure 15.1, counting from top picture to bottom picture, which picture reflects the conversation of response to that I've documented in your notes at this time? Which picture is it? Okay, thanks everybody for responding here. Yeah, I appreciate that, and I want you to know that it is not picture 3. It is not picture 3. Okay, so let me ask you in this. For our current conversation about response to, Hold on a second. I can't type worth a darn here. Here we go. For response 2, is the presynaptic neuron going, is it connecting to the adrenal gland? For response 2, is the presynaptic neuron connecting to the adrenal gland? Okay, it's not. It is not. Okay, so please reconsider your answer to my first question. Okay, which picture, top to bottom, are we currently talking about for response 2? Remember, we're counting the pictures top to bottom. Okay, top to bottom. So which picture is describing our current conversation of response 2 in the notes? We've established it's not picture 3. Picture 3 is counting from the top, right? Okay, all right. Okay, so take a look at your chat window, folks. See what people are answering right now. Sierra and Christian, this is not picture 4. So picture 4 would be the bottom picture, correct? It's not picture 4. Sierra and Christian, do you see that picture 4 at the bottom is involving the parasympathetic nervous system? And we are not in the parasympathetic nervous system right now. We are in the sympathetic nervous system. So the only logical conclusion is picture 2. Picture 2 of figure 15.1 is the answer to my original question here about response 2. Response 2 is illustrated by picture 2 in figure 15.1. Again, picture 2 counting top to bottom. Okay. Is this slowly making sense, folks? Do you see why it's not picture 4? And do you see why it's not picture 3? Okay. This is one of those things where you really got to sit and stare at it and make sure you know what you're really thinking about. Okay. So response 1 that we put in your notes, response 1 about the adrenaline release from the adrenal gland, that's picture 3, counting top to bottom. That's picture 3 of figure 1. Response 2 is picture 2 of figure 15.1. Okay. All right. I know this is not easy. This is not easy. This is not trivial. And think about it, the next time you go to a nervous system specialist, what you're asking him or her to do for you. This is not easy, okay? All right. Okay, gang. So any questions right now? We're starting to get short on time, so I need to go a little faster, but anything you'd like to ask about now? Okay. And so my goal in going through this here with you in notes during class is so that when you see my challenge to you in the study guide for the final exam, you don't melt and just give up, because I'm going to ask some pretty tough stuff, but I'm trying to model right here, right now, the manner in which you need to think to answer my questions and prepare for the final exam, okay? All right, so let's continue here. So we've now described two responses to fight or flight. Now let's switch gears and say, okay, I've now run out of the auditorium. I've run away from Frankenstein and I'm outside in the sunshine and I see other happy people around smiling at me. So I don't need to feel afraid anymore. So how does your body respond after the fear stimulus has gone away? Once the threat has been removed or once the threat has been avoided, how does your body calm down? Okay, so here now, let's tackle this. After the danger or threat has passed, how does your body calm down? Well, you can appreciate that this is now going to involve the rest and digest system, which is the parasympathetic subdivision of the autonomic nervous system. Okay, let's tackle this, okay? And so now I'm going to go a little bit faster, folks, okay? Response number 3. I'm going to keep the responses, numbered 1, 2, 3, and 4. Response 1 is part of the fear response. Response 2 is part of the fear response. Response 3 is going to be part of the calming down response. Okay, so this is after the threat has passed. So after the threat has passed, the hypothalamus is going to send nerve signals. It's going to send electrical impulses down different axons of different neurons that are categorized within the parasympathetic subdivision of the autonomic nervous system. So let me ask you in the chat window, which picture of figure 15.1 illustrates the parasympathetic division? Okay, Christian, you're absolutely right. It is picture 4, the bottommost picture there, right? When you look at the words, you see parasympathetic. Okay, so I'm sorry, I'm probably going to run out of time here. We're probably going to need about 20 minutes next class before I can start chapter 16, but that's not too bad of a problem. Okay, let's keep going here to try to finish this. So for response 3, you're going to get nerve impulses down neurons that are part of the parasympathetic division. You're going to have axons leave the brain stem above cervical vertebrae C1. So for this, I want you to turn to figure 15.3, figure 15.3. Figure 15.3, okay? And let's get this into your notes. Axons leave the brain stem above cervical vertebra C1 as part of the facial nerve, right? You're learning the facial nerve for lab, right? So take a look in figure 15.3 and identify the facial nerve. Identify the facial nerve. Okay. The facial nerve synapses with other neuron cell bodies that are part of the submandibular gland. Okay, so you can see that the facial nerve would contain neurons, which are presynaptic neurons, which synapse with other neurons in the pterygopalatine ganglion. My God, look at that word, okay? So there's a synaptic connection there. The postsynaptic cells are depolarized. They send an X potential back into the salivary glands to reverse the effects of dry mouth. So I want to finish this, folks. If you can hang with me for 2 minutes, I appreciate it. So axons leave the brainstem above cervical vertebrae C1 as part of the facial nerve, and synapse with other neurons, okay, that are part of two ganglions there. I mentioned pterygopalatine. If you trace that out, you're going to see some of those postsynaptic neurons go to the sublingual and submandibular glands. But we also have connections with the submandibular ganglion. And take a look there. You also have a connection. Well, it's getting real complicated now, isn't it? It's like a spider web. So let me simplify this. Part of the, I'm going to say the submandibular ganglion right now, and other ganglions that enter the salivary glands. Axons leave the brainstem as part of the facial nerve and synapse with other neurons that are part of the submandibular ganglion and other ganglions that eventually enter, let me say it this way, whose axons eventually enter the salivary glands. Okay. So within the salivary glands, the postsynaptic cells coming off of these ganglions, they're going to release acetylcholine, which causes the arterioles in the salivary glands to dilate, which is to say widen. So the blood vessels in the salivary glands are responding to acetylcholine released from the postsynaptic cells coming out of these ganglia, themselves of which were activated by presynaptic neurons of the facial nerve. In the salivary gland, the acetylcholine causes the arterioles to widen, open up, which restores the flow of blood into the gland, which allows the gland to filter the blood plasma and increase its production of saliva, meaning we effectively relieve our dry mouth situation. Okay, I know that was fast, a little bit more fast than I would like it to be, but hopefully you get the basic idea here. Okay. All right, folks, I'm over, so I need to stop. I'm going to stop here. I'm going to ask, do you have anything you'd like to ask? So when you start looking at your final exam study guide, start thinking about how we've talked about these ideas during lecture time. as you begin to prepare to answer my questions on the final exam. So there's a question in the chat window. Will we be meeting for lab on Tuesday? No, you will not be meeting for lab on Tuesday. You will be taking your lab exam, and that is it. Okay? Okay.